Now let's talk about, I'm going to leave it here, I'm just trying to make shorter bits. So let's talk about B cells, okay? So B cells mediate humoral responses, okay? Now, why is the term humoral? Humor in Greek means fluid. The responses that B cells produce are antibody-mediated responses. Am I clear? These antibodies are going to be found in the blood, in the fluid. Therefore, humoral. Clear? Good? Okay. So B cells originate in the bone marrow. For that matter, every cell that is found in the circulation, every red, white, and blood cell and platelet, they all ultimately originate in bone marrow. Okay? And then they remain in the bone marrow for the maturation. They go to school, so to say. So imagine like you're born in Columbus and you go to Ohio State. Okay, you're not getting out of the city. Now, what happens after the maturation? They seed lymphoid organs. Now, do you understand the term seeding? Okay, so seeding refers to the fact that B cells move into your lymphoid organs and stay there. Lymph nodes, spleen. Does that make sense? Are we clear? Now, I'm not going to go down this rabbit hole. I just want to say that immunology is full of exceptions. So basically, if you ask me, are there situations when B cells are not in lymphoid organs? The answer is yep. But for our intents and purposes, we're going to focus on this. Okay? Does that make sense? Now, let's take a look at what happens in those lymphoid organs. So imagine that now, my further drawings are going to be incorrect from the standpoint of structure, but my goal is different. I want to show something different. So imagine you have B cells. So I have 3 clones of B cells. So those are clones. Clone 1, 2, and 3. Clone 1 expresses antibody, check this out, of this specificity. Clone 2 produces antibodies, can produce antibodies of this specificity. Clone 3 can produce antibodies of this specificity. Do you see the difference? Now, question, do they produce antibodies, those B cells? No, not yet. Now look, check this out. These B cells encounter an antigen. Okay, this is antigen. I have a question. Which of these 3 clones will interact with an antigen? Take a look. 3, of course, because it has a specificity, right? So this moment when antigen interacts with a proper B cell clone and, look, selects it, it is called clonal selection. Does that make sense? After the clonal selection, what happens to B cells? That clone, and only that clone, will differentiate and proliferate. Remember, differentiation is the term reserved for cells becoming more specialized, while proliferation refers to cells becoming more numerous. Does that make sense? We're good? So what do these cells differentiate and proliferate to? They will differentiate into memory B cells. Okay, they will keep immunological memory about this antigen. And they will also differentiate and proliferate into plasma cells, which will produce a whole bunch of antibodies with this particular specificity. Does that make sense? So again, after the clonal selection, B cell differentiates into memory B cells and plasma cells, and it proliferates. So these plasma cells and memory cells become more numerous. Are you with me so far? Now, this process of differentiation and proliferation of immune cells is called clonal expansion. So this process is clonal expansion. Does that make sense? We good? Now I want to highlight a couple of things here. First of all, how B cells do not recognize self-antigens. Okay, if you think about it, listen to me. All this breadth of B cell specificity should result in the fact that, there will be some B cells that can recognize human proteins, right? How do we manage not to attack our own antigens? Turns out that when B cells go to school, they undergo negative selection. Negative selection is the process in which self-reactive B cells undergo apoptosis. This is what essentially happens in the bone marrow. B cells that can otherwise attack our own human tissues instead are sent into an apoptosis. In an ideal case. Are we clear? Good? Questions?
Do the antibodies that plasma cells create, those ones, do they not need to be attached to a cell to, like in the other picture there?
Here?
Yes. Is that antigen, or the antibody, is it connected to
A cell? No. No. This is why, it's a good question. This is why it's called humoral. Antibodies are intrinsically, at the beginning of their work, they are soluble. If you pull down, let's say I'm going to go and get my booster shot, I don't know, today. In a few days, you can take blood sample from me, pull down plasma, just clear formed elements, get the plasma, and you will be able to detect soluble antibodies. You see what I'm saying? This is why, for example, one of the ways to try, attempted ways for the antiviral, anti-coronavirus therapy was a plasma transfusion. Blood transfusions technically. Take from a person who was sick and recovered and take this convalescent plasma and inject into a person who is actively sick. It didn't work for multiple reasons, but we're not going to touch on those reasons today. Okay, so far, does that make sense? The process of clonal selection and clonal expansion.
That bottom part where those antigens, those three that kind of just come off, yeah, that.
Those are antibodies. Plasma cells just produce them.
Why there do they just go down and then up there they connect?
Oh, yeah, that's what I was talking about. So, plasma cells have that unique capacity to produce soluble antibodies. Only B cells, they are connected, they serve as a cell receptor. So, this cell cannot make antibodies. It has to become plasma cell. B cell has a potential to make antibodies. Which ones? The same as the receptor on the surface. So essentially, the receptor is the non-soluble antibody. Okay? Does that make sense? Okay, we got this. I'm trying to think about something. Oh, yeah. One important feature of B cells that I'm going to mention verbally. If you look at my depiction, I show you that B cells can directly interact with antigen. It is true. Partially. They can do that. In about 20 minutes, I suppose, we're going to talk about T cells. We will figure out that they do it much better when they have a help from T cells. Does that make sense? Okay, we're good? Any questions? Okay.